Hello, I'm Andy Zhang, and this is my presentation for Scheme OSPREY, which is my project in the Donald Lab as part of CS+. So let's get into some rationale and structure of what Scheme OSPREY is. Scheme is a functional programming language in the Lisp dialect of languages, and it supports things like higher order procedures and produces elegant and reliable code, which is minimalist and easy to read. On the right side, we have a little diagram of OSPREY. OSPREY 3.0 is our laboratory's protein design software package. And what it does is that what you would currently do to write an OSPREY script would be you would write a script in Python, and the Python would go through this package called jpipe to call the Java backend code in OSPREY. Now we could do something similar like that for our Scheme front end. But it turns out that GNU has a software package called Kava, which is Java written in Scheme. Which is Scheme written in Java. So what you do now with Kava is that it's written directly over top of the OSPREY backend code, which is Java. And that eliminates the use of a middleman like jpipe, which allows us to just call Java code directly without having to interface between languages. So to write this interface, we need to import and write wrappers for all the relevant OSPREY classes and allow the Scheme frontend to access any relevant fields and create the objects it needs to create. Particularly tough things to do when converting between Scheme and Java were to implement OSPREY interfaces in Scheme. So some of the interfaces in OSPREY were Java classes, and we wanted to be able to create some kind of Scheme class that could inherit a Java class, which was pretty difficult to do. Another thing we needed to do was to keep track and coerce types. So Scheme isn't very strongly typed, which means that keeping track of what Java classes you're passing in, what types you're passing in, is pretty difficult. And also in Scheme, there are no doubles or integers. They're all just numbers, which means that when we're passing in some Scheme numbers to Java code, we need to ensure that they're either doubles or ints or they're represented correctly. And lastly, we needed to minimize side effects. So if you've used Java before, you know that Java, you can set variables very easily. Whereas in Scheme, it's a constructive language. So rather than setting variables, we would rather create a new object with the relevant fields updated. So we don't actually go into an object and change fields. So some first steps to testing out this interface. We wanted to translate some Python scripts into Scheme and test out if all of the wrappers are functional. So on the left, you can see there's some code written in Python, written in Scheme, translated from Python. And you can notice that there's this code isn't particularly Scheme-like. If you look at the right, we have the same code in Python. And if you look between those two code segments, they're pretty much identical, other than syntax in Scheme versus syntax in Python. There's no really, we don't really take any advantage of what Scheme has to offer. So there's no functional programming. And most of this is just a Python direct translation. And that's not really what we want to do if we create a new interface in a different language. So after an iteration and consulting with people in the lab, we created this segment of code on the left. So as you can see, we take advantage of higher order procedures. You can see that we use map. And this map allows us to abstract the code where we prepare the protein. And instead of having to edit different lines, we can now just use this code and change what the map is being applied to. Namely, we only need to change different numbers in the make residue section here. Pardon me. And also change what amino acids they can mutate to down here. And you can also notice that any extraneous representations in classes we don't need are now internal representations within our import file. So you can see that there are no strings anymore because we don't need them. And all the strings have been migrated into the import file. So we want to create also an IDE, so that way you can code easily in this interface. So one idea was to create an interactive REPL via Emacs. Now this would function similarly to Racket or Dr. Racket if you've used that in CS 230. And what it allows you to do is to code, run the code, and in the interactive panel below, it has all the definitions in the code block above saved, and you can code even more in the section below. This allows you to type something in and load in your relevant script, and then you can run it and then define everything you need to define and type in more things to run below using those definitions. Another option would be to use a shell script with the appropriate class path. You can see below that the class path you would have to manually invoke is quite long and tedious to write. So a shell script is pretty welcome. The only issue here is that you can't really interact with it. So if you type in a class path and want to run a file, you can't add on anything after you run the file. It will terminate after it runs the file. If you don't run a file, you can type in the file manually. So the interactive REPL really combines these two features of the shell script, which is very helpful. So then after everything was done, we needed to convert all of the example scripts in Python. And as you can see, there are 5 extensive folders of Python example scripts into Scheme. So we translated these to Scheme, we ran them to make sure everything was working, and then we schemified them in the same way we did so for the first example script. Then after that, extensive documentation was written for all of the wrapper functions and all of the example scripts. But now that the interface was pretty much done, we wanted to put the Scheme frontend to use. We wanted to make sure it could actually be used in real designs. And the goal of this was to achieve 2 objectives. One of which was to verify that older designs and early OSPREY versions could also work in this new version, and also ensure that actual designs could be done in Scheme. So to do so, we tested this on some HIV designs that have also been tested in the wet lab. And on the right, you can see that there's this picture of an HIV protein that we're targeting. And for those who don't know, HIV, how it infects the cell is that it binds to the cell membrane and then enters this fusion state. So this is a protein in question that binds to the membrane. And we want to find out some way to prevent it from binding. So what is the design? What we do for a design is that we want to find a protein or some residues that are important to binding. More on what residues are later. And we want to see if we can make a new protein or redesign that protein to be better at binding. So that way it doesn't bind, instead of binding to one of our cells, it binds to this protein. And what a protein is just it's a sequence of amino acid residues. And we want to find the residues that we want to mutate and choose what amino acids they're mutable to. Then we want to select what residues are flexible. So they can move around, but they can't change the different residues. Typically, we want these to be close to the mutable residue. So that way, if we change from a smaller amino acid residue to a larger one, we're able to move the other ones out of the way so we don't get any steric clashes. And afterwards, we want to calculate binding constants. On the right here, you can see that we select a mutable residue, which is in orange, and everything around it that is flexible is in blue. And looking forward to next steps, we want to apply the Scheme frontend to other meaningful designs, such as the COVID-19 epidemic that we're currently in. And we also want to allow students who took CS 230 to easily redesign proteins in Scheme, which allows for more people who are interested in protein design and have taken some functional programming before to be able to really use what all this interface has to offer. And we wanted to be able to code elegant programs in Scheme with actual biomedical applications. With that, thank you for listening to my presentation.